So welcome everybody from Cafe. We're the BU and Harvard Climate and Health Research Coordinating Center. Today's session is going to be a little bit different from our usual webinar format. Today, we're hosting a panel on a particularly intriguing topic, environmental pressures and fungal disease epidemiology. And our panel today is going to be led by two experts on climate change and disease, excuse me, Dr. Alexandra Heaney, who is from the University of California, San Diego, and Dr. Jennifer Head, who's from the University of Michigan. Thank you so much, Dr. Heaney and Dr. Head, for hosting this session. And I'm now going to turn it over to you all to introduce the session and the panelists.
Thank you so much, Rachel. I'm going to share my screen. Okay, does that look okay? All right, great. All right, well, good morning, everyone, or good afternoon, depending on where you are. Thank you so much for that introduction, Rachel. I am Alex Heaney, an assistant professor in the Herbert Wertheim School of Public Health at UC San Diego. And my co-moderator for our session today is Dr. Jennifer Head, assistant professor of epidemiology at the University of Michigan School of Public Health. It is our pleasure to welcome you to today's panel on how environmental pressures alter fungal disease epidemiology. So today we're going to discuss how environmental change is driving new patterns in human fungal infections. The distribution of many fungal pathogens has historically been tied to local climate and soil conditions, but now rapid changes in those conditions are redrawing the map of infection risk and really challenging our existing tools for control and treatment. Extreme heat and changing weather patterns are enabling pathogenic fungi to adapt to warmer temperatures, expand into new geographies, and in some cases, become more virulent. We're also seeing increased use of agricultural fungicides in response to food insecurity and crop pressures, which has implications for antifungal drug resistance. So our goals today in this panel are first to spotlight these shifts and highlight what we do and don't know about how environmental change is influencing fungal disease patterns. And two, to discuss how to translate this knowledge into public health action to prevent and mitigate future fungal disease burden. So we've brought together 7 fantastic leading experts from across several disciplines, spanning epidemiology, microbiology, mycology, environmental health sciences, veterinary science, and state and federal public health to address these questions. We'll begin with a series of short talks, each about 5 minutes long, where each panelist will introduce a piece of this complex puzzle. After that, we'll move into a moderated discussion and Q&A. We already have some questions prepared, but we would love to bring your voices into that discussion and Q&A as well. So please feel free to post your questions in the chat as they come to mind. We'll keep track of them and then revisit them at the end. Okay, so with that, our first presenter is Dr. Asiya Gusa, assistant professor of molecular genetics and microbiology at the Duke University School of Medicine. Dr. Gusa's laboratory examines how heat stress drives thermal adaptation and antifungal drug resistance in pathogenic fungi. She's also a founding member of the CLIF Climate and Fungi Research Group, which is currently investigating health risks tied to indoor mold growth after flooding events. Thank you so much for being here with us, Dr. Gusa. I will hand it over to you. And let me get my slides.
Awesome. Thank you so much, Alex and Jennifer, for inviting me to be part of this. And for all of you listening there, I'm looking forward to the discussion. So yes, as Alex mentioned, I'm looking at heat stress and thermal adaptation and drug resistance in pathogenic fungi. Next slide. So I wanted to just draw your attention to, just as Alex was explaining, some of these environmental pressures that we have, that fungi are facing in the environment. And we're very much interested in the impacts on human health and fungal disease in my lab. And I'm just showing you here an image from a book chapter that we just published, which is available. And you can see that we're looking at the disruption of how the disruption of fungi in soils results in the dispersal of spores into new geographic regions. This is contributing to the spread, and we're starting to see changes in biodiversity in our soils. We can also see the impact of heat stress through wildfires and also increased global temperatures. And we really do believe that these are creating a selective pressure for fungi to evolve to grow better at higher temperatures. Next slide. So in our lab, we study stress adaptation and fungal disease. And in particular, we focus on a pathogen called Cryptococcus, which we inhale in the air into our lungs. It can cause an infection there or spread to the brain and cause a fatal cryptococcal meningitis. What we're interested in is how these environmental fungi that encounter these stresses in the environment, do they actually have mutations that occur that cause them to adapt to either cause further infection or persist in the human body? So we're interested in the impact of climate change and also the impact on human health. Next slide. So in particular, we've been focused on heat stress and looking at the impact on mutation rate. We want to know, are these mutations that arise, do they actually result in adaptive pathogenic traits in fungi? So for example, we're looking at the impact of heat stress in the environment. Is this resulting in the evolution of thermal tolerance among those fungi that are less virulent or even non-pathogenic? And we're also interested in that transition from the environment to the human host and the heat stress it encounters there. Does this result in the evolution of drug resistance and increased virulence during persistent infections? Next slide. Some of the key questions that we're asking are, how does heat stress affect mutations in pathogenic fungi? And also, what mechanisms are driving heat adaptation? Next slide. So to do this, we've been focused on what I call sister species of Cryptococcus, which are very closely related. On the left, you'll see Cryptococcus neoformans, which is the major disease-causing strain species that actually results in the majority of worldwide infections. It's more thermal tolerant and more deadly compared to its sister C. D. neoformans. And so we're sort of able to compare these two. Next slide. As we were first doing studies in Cryptococcus D. neoformans, the less thermal tolerant, we came across this striking finding that if you grew cells under heat stress at human body temperature, or 37 degrees, compared to 30 degrees, we saw a major increase in the drug resistance rates across different antifungals. And this was what was even more surprising, was that the cause of that increase was due to transposon mutations, these mobile genetic elements that would jump into gene sequences and cause increased drug resistance. We've since confirmed that this phenomenon happens in both sister species of Cryptococcus, and we're investigating it further. Next slide. So transposable elements were first discovered by Barbara McClintock, who won the Nobel Prize for this. And at that time, she actually predicted that environmental shocks could stimulate the mobility of genetic elements to restructure the genome and foster adaptation and evolution. And we're seeing more and more evidence of this in recent publications. Next slide. So one of the key questions that we're looking at is, what is the mechanism of thermal adaptation in Cryptococcus? So as this yeast encounters higher heat temperatures, such as body temperature, where it doesn't grow that well in, if adapted over time, we're looking to see what adaptations occur. And what's really striking is you can vary in a very short amount of time or passage of the yeast, you can actually see these adapting fairly quickly. And so we're looking at the earliest time point where thermal tolerance evolves, and then doing whole genome sequencing to determine what mutations are actually there causing the effect. Next step. So we've already identified some genes of interest. And what's really has been remarkable, at least in the strain that we're studying, is that all of the candidate genes appear to have transposon mutations in either uncharacterized genes or genes that have been characterized that seem to be causing the thermal tolerance. So beyond what's causing the mutation, we're also interested in whether or not this thermal adaptation actually results in a fitness gain for virulence, or is there potentially a fitness loss for pathogenicity? So that's one of the questions that we're very excited to answer next. Next slide. And finally, I just want to say that we're beginning to look at emerging fungal health risks in the aftermath of flooding. So we know that there are these fungal blooms, a lot of mold that pops up after natural disasters such as hurricanes and major storms, which we're seeing ravaging the country at this moment in time. Next slide. We've been looking at Hurricane Helene, which really recently hit in September 2024. It hit Western North Carolina. You can see some of the affected areas here. Next slide. And some of the water levels and the indoor tracking of mud into people's homes and places of residence and business. Next slide. So we've been doing sampling and growing what fungi are growing in the environments and asking the questions, what kinds of mycotoxins are they releasing? What kinds of impacts are they having as far as fungal infections? Or is it more associated with exacerbating existing respiratory conditions? Next slide. So I'll just wrap up by thanking my folks, and one more slide. And just announcing that at Duke University, we just had our inaugural Fungi in a Warming World, Adaptations, Changes, and Resilience. It's a fungal symposium about climate change. And we're going to have another one May 29th, 2026. So stay tuned for that. Actually, Jennifer Head is one of our invited speakers for that. So well, thank you very much. Thank you for your time. Look forward to the panel discussion.
Thank you so much. All right. Thank you so much, Dr. Gusa. That was such a wonderful start to our panel. Next, we are welcoming Dr. Martin Hönigl, Associate Professor for Translational Mycology in the Division of Infectious Diseases at the Medical University of Graz, Austria. Before returning to Austria, Dr. Hönigl was Associate Professor of Medicine at the Division of Infectious Diseases and Global Public Health, actually at my institution, UC San Diego. With more than 15 years in clinical mycology and over 350 peer-reviewed publications, his work spans fungal diagnostics, pathogenesis, and antifungal stewardship. I will hand it over to you.
Thanks so much, Alex, for the very nice introduction. So yes, very happy to be here and a very exciting panel. So let's get rolling. So I'll give a little bit an overview. My background is infectious diseases, actually, as Alex mentioned, and I'll give an overview of kind of like all the different impacts. We heard a little bit about resistance, but there's multiple other impacts as well. So let's go to the next slide, please. So one key question that we are asking, of course, ourselves constantly is when we compare ourselves as humans to many animals, and especially, animals that are colder, reptiles, for example, that why is it that fungal infections are not yet posing a bigger threat to us humans? And we know that fungi are ubiquitous in nature, found in and on our bodies. And there's much more fungi in the world in terms of mass than actually there's human mass in the world. Nevertheless, of the estimated, and this estimate is really rough because it changes all the time, basically, but over 5 million species, only a very small number of about 300 are known to cause disease regularly in humans. So still, we seem to be resistant to most kind of fungal infections. And the number one reason probably, and I think Dr. Gusa jumped into that as well, is that we have the warm temperature and we eat a lot actually to keep our warm temperature. So it kind of like, directs our life. But this is really saving us from many of the fungal pathogens. And of course, now if the world is getting warmer and fungi adapt to warmer climate, and at the same time, the humans actually are getting a tiny bit colder every decade or so, these temperature differences may get smaller in the future. And I think important is for fungal pathogens always to remember that, they are exposed to heat stress as well. So not all of them are thriving, but those who remain and those who are thriving are not only exposed to heat stress, but also exposed to multiple other stresses that come together with heat stress, like, for example, stress due to increased UV light exposure, where they also develop resistance mechanisms and mechanisms to deal with that stress that on the long term can harm us humans. Let's go to the next slide, please. So if we look here a little bit further and look into the impact of climate change on the fungus, one important topic, of course, is development of dermatolerance, which then may lead to the emergence of new pathogenic species for us humans because they get used to the human body temperature. And one potential example for this, and there's other components as well that contributed, is of course the emergence of Candida auris. But then there's also the increased virulence that fungi learn in response to the climate change stresses. That then, of course, causes a bigger harm to kind of like agricultural products, food production, leads to increased use of fungicides in agriculture, and thereby drives resistance, for example, by killers. Dispersal, obviously, natural disasters are doubling, so we see more dispersal after wildfires, but also through all wind-associated natural disasters where fungal spores can disperse, and geographic range is changing, which is, I think the most obvious example is cocci, which we really have seen spreading over the last decades due to changing climate, drier and warmer climate. However, there's also an important impact on the human host, and that's where especially also the natural disasters come in. But let me focus a little bit on resistance, because that's obviously for me as an ID guy is something that we are seeing in our patient. Next slide, please. That's something that we are also seeing increasingly in our patients. Yes, this one. And I think the main problem here is obviously that this whole topic, we are always tending to challenge, to work on this topic only from one side, which is the human disease side. But I think it's very important, the one health approach, one world, one health approach here. Because obviously fungal pathogens are the most important pathogens threatening food production as well. So on a worldwide level, this is maybe as important or even more important for us humans than the cases of diseases that we're currently seeing. But of course, both are important at the end. So we have to find a way together with, the bodies that focus on fungicide, approvable fungicide use, etc. In the environment to find a solution that fits both sides. And the problem, of course, with basically the with the resistance is that the fungal pathogens have developed increased virulence and are increasingly a threat for our food production. That's why we have to use more antifungals in the environment. And this is not only on the crops, but it's also already in the earth around on the fields. And then fungal pathogens in the plant pathogens develop resistance to that. So we need newer antifungals to be used as fungicides. And this leads to the crazy situation currently that we have now fungicides approved for use that contain pretty much similar antifungal substances than the newest antifungals that we are currently developing for human use that are still in phase 3 trials. So we have these new antifungals first in agriculture before we can even use them in humans. And this leads to development potentially of resistance before we even can use them in humans. Next slide, please. Of course, another big topic is the natural disaster part. And here, I think it's very important to emphasize that natural disasters with climate change have doubled in numbers. It's not just more in the news, they really have doubled in the numbers over the last 2 decades compared to the 2 decades before that. And there's numerous ways how basically natural disasters can contribute to fungal disease outbreaks in humans via traumatic injuries. We heard from Dr. Gusa about dispersal, but also, of course, we heard about the aftermath of natural disasters, sick housing, moldy housing that can lead to fungal diseases in the long term. There's examples for all of this basically in literature, but I want to kind of like emphasize one more point in my presentation. I think that's very important to consider when talking about climate change and fungal diseases. And please, next slide, Alex. And this is basically that there's other factors as well that drive fungal diseases. And one of the factors, or some of the factors, are the social determinants of health. And they have really been under the radar for a long time because fungal diseases for us is always something, this is increasing with, as opportunistic infections with advances in medicine. But on a worldwide level, and we looked on this also with Jeff Jenks, also from UCSD, old colleague from UCSD, basically, where we looked into this and also with support of WHO, if we can go quickly back and looked into all the different social determinants of health as defined by WHO, and found basically fungal outbreaks associated with all of them, whether it's exposure for working conditions, poor housing, basic amenities conditions, substance use and crime, but also, of course, structural conflict that may contribute to fungal disease outbreaks. And all of this basically leads, and now please next slide, that of course the social determinants of health we are talking about are much more prevalent, not in the richer countries of the world, but in the low and middle income countries. In those countries where we have more limited antifungal diagnostics, more limited antifungal treatment, where we have restricted training of healthcare professionals, and of course have not even the possibility to diagnose or report these infections. And that's important. But when we are seeing something here in the US or in Europe, this is only the tip of the iceberg. On a worldwide level, the whole situation is much faster worsening in other parts, where we really don't even have the tools to cover this worsening. Next slide, please. Alex, can we just go next slide? Yes, and this basically brings all together. So we're living in times where we know that the social determinants of health, together with industrial factors, one slide back, please, drive basically climate change and the global warming. Alex, can we go one slide back? Yes, here, perfect. But then we also know that climate change by itself drives, again, the social determinants of health and inequalities. So it makes this worse. So this is a vicious cycle that we're currently in, and both contribute to antifungal resistance development and emergence of new fungal pathogens. We also have, of course, increased natural disasters, and all of that together is impacting medical mycology. And important to emphasize, it's impacting it worldwide. So it's not a problem that's reduced to our country. It's really a worldwide problem. And with that, thanks so much for having me, and really looking forward to the discussions. And last slide is just my team, my key team at the Medical University of Graz. And thanks.
Wonderful. Thank you so much, Martin. Sorry for the weird slide things. The slides had a mind of their own. But that was a really wonderful overview and of all of the work you've done to think about the sort of concepts and frameworks in this field has been fantastic. Let me get back to our... Well, okay. Mandy, you won't get a picture introduction, but our third speaker is Dr. Amanda Weaver, who earned her doctorate in environmental health sciences at UC Berkeley, examining the socio-environmental epidemiology of valley fever. She now serves as a research scientist in the climate change and health equity branch at the California Department of Public Health. Dr. Weaver, welcome.
Awesome. Thanks, Alex. Ready whenever you are. Great. Okay, well, so thank you to the panelists who went before me. I think it was an excellent setup. I'm going to be kind of zooming in on California and valley fever, talking about some of the emerging environmental pressures on mineral dust exposure and how that intersects with valley fever. So you can go to the next slide. So coccidioidomycosis, just a bit of background on it. It's an emerging climate-sensitive fungal infection that is caused by the inhalation of spores of the fungus Coccidioides. As you can see on the right here, when the dirt and dust are disturbed either manually or by wind, that's when people inhale the spores and become sick. And so the symptoms are quite similar to pneumonia, which can make diagnosis a bit difficult, but they include things like headache, fever, night sweats, and rash, and dissemination, which is the term for when the infection spreads out of the lungs to other parts of the body, like the bones or brain, is really quite rare, but very serious. And chronic coccidioidomycosis is also a really burdensome disease for patients, both financially, emotionally, and physically. Next slide. And so on the epidemiology side, in California, we've seen over the past several decades or so that incidence has been increasing in and around the San Joaquin Valley, which is the traditional endemic zone. But kind of overall, you see this positive trend, which has continued in the years past 2019, But I'll just draw your attention to the fact that the fastest increases since the year 2000 have been in the northern part of the San Joaquin Valley, and in more recent years on the coastal counties of California. So again, sort of what other people were mentioning, this expansion outside of the traditional endemic regions has been noted. Next slide. And so for coccidioidomycosis, there's sort of these 2 to 3 levers that environmental pressures and sociological pressures really act on that relate to the growth cycle of the fungus, namely the mycelia require water to grow. So the rainy season really supports this phase of the life cycle. And then as the mycelia become arthoconidia, increased heat supports sort of the aerosolization of both soil and spores, which is how people get exposed. Next slide. Oh yeah, you can pass those. So just to sort of quantify this risk, previous studies had really focused on the risk that's associated with exposure to all source particulate matter, but we wanted to zoom in a bit more on dust exposures, which is the co-exposures between soil and spores. And so working with the State Department of Public Health, we were able to conduct a time series analysis to assess the risk posed by exposures to dust. And we found that kind of combining the incubation period and reporting delays that are inherent in the healthcare system, that dust exposures occurring into the 1 to 3 months prior were most strongly associated. You just click the next button. And so this 1 microgram per meter cubed increase was associated with around an 8 to 15% increase in incidence. Go to the next slide. And so when we have these sort of different levers of precipitation, heat, and dust, this is sort of where these socio-environmental pressures really impact dust exposure and risk of coccidioidomycosis. So as a previous panelist mentioned, sort of underappreciated mechanism is our occupations and the social determinants of health. So major soil engaged occupations in California include construction, agriculture, and wildland firefighting. And we know under climate change, we've seen over the past decades and recently that the wildfire season has been expanding and increasing in intensity. And we've also seen occupational outbreaks among this particular occupation, as well as big increases in construction and agriculture demands, which are 2 highly soil engaged occupations. Then sort of flipping to the extreme heat side, I think there's been some interesting discussion about adaptation that I think, us in the valley fever world would really benefit from kind of diving into a bit more. I think also we've seen that these extreme heat events have, just from the heat related illness side, have been having the biggest impact along the coast of California because they've been historically cooler. But this is sort of an area that overlaps with the more recent increases in incidence that we've seen over the past couple years. Next slide. And so then also on the precipitation side, we've seen trends over the last 6 decades or so where the precipitation or rainy season has really been decreasing in the fall, particularly November precipitation being quite low, and a delay of around a month that has led to this sort of sharper, more extreme rainy season. And kind of interestingly, in the last couple of years of 2023 and 2024, when the state has its provisional data available, we've seen that there are a relatively high number of cases and peaks later in the year than are previously recorded. So, sort of historically, the messaging has been late summer, early fall, and in these past years, we've seen these peaks around November, December, January, which I think is kind of an interesting recent phenomenon and maybe to do with some of these shifting pressures. Next slide. And so kind of lastly, I just wanted to end on this note of, in the absence of the vaccine, the environment, both the social and natural environment are really the levers that we have to be working with to protect people. So kind of this increasing clarity that research has been giving us on how these climate cycles impact the peak and the burden of where and when and who are being impacted has been very helpful in tailoring warnings to healthcare providers. And sort of this next step is really reaching the general public. But again, dust control is really typically highly localized and short term. These occupational interventions like wearing a mask are super infeasible for people who are dealing with all sorts of other environmental hazards, most pressingly extreme heat. And so I think this sort of area that we need to be moving towards as these regions shift is advocating for patient and provider awareness, especially as this region expands. We have to move past our sort of traditional idea of the San Joaquin Valley and put these resources into the places that are popping up for us in these recent years. Next slide. Great. So thank you so much for listening, and excited for the panel part.
Wonderful. Thank you so much, Mandy. That was a wonderful summary of all of the great valley fever work that you and our group has been doing for the last several years. Go back to our slides here. Okay. All right. So our fourth presenter and my co-moderator is Dr. Jennifer Head. Dr. Head is the John G. Searle Assistant Professor of Epidemiology at the University of Michigan School of Public Health and a core member of the Institute for Global Change Biology. Dr. Head applies mathematical and statistical models to understand the role of hydroclimate extremes and zoonotic host behavior on fungal infections. Her work integrates data from surveillance systems, serological and skin test surveys, and field studies. So Dr. Head, I will hand it over to you. And I believe you are going to share your screen. So I will stop sharing.
Awesome. Thank you so much. Are you seeing my slides?
Yes.
Yes. Okay. Thank you so much. Well, thank you, Alex, and all the other panelists and viewers for being here today. I'm really excited to talk a bit more about hydroclimate volatility and fungal diseases. And I think Dr. Amanda Weaver set us up very nicely talking about valley fever, because much of my talk will concern valley fever as well. So first, so what is hydroclimate volatility? You may have heard hydroclimate volatility referred to as precipitation whiplash, but it's this idea that wetter conditions under climate change are getting wetter and drier conditions under climate change are getting drier. And we can understand this if we think of the atmosphere as a type of sponge. So as the atmosphere heats up, its water vapor holding capacity increases. So in a sense, the sponge that is the atmosphere is growing in size, which means that it has greater ability to absorb water through evapotranspiration, but it also has greater ability to release water when you can think of that sponge being wrung out. And this phenomenon is happening pretty much globally, but including in the southwestern US, where we see high rates of valley fever, where a 25 to 100% increase in extreme dry to wet precipitation events is projected under climate change over the coming century. So if we think about the life cycle of many pathogenic fungi that live in environmental media and are inhaled by human hosts, they actually require the completion of multiple disparate processes. So first, these fungi grow as long branching chains in the soil, which is a process that requires moisture and nutrients. Those chains then sporulate. Those spores are released through some sort of breaking mechanism. And then those individual spores can become airborne, where they become inhaled by a human host. So that latter process, that becoming airborne, is really facilitated by dry conditions. As the soil dries, the soil becomes more erodible and spores can become more easily aerosolized. So within the course of a year, wet and dry cycles are really supporting both that growth process as well as that dispersion process. Now, wet-dry cycles can also happen on a multi-year scale, and it can be easy to think of this in the context of the southwestern US and California, where we might have multi-year droughts punctuated by years with extreme atmospheric river conditions. And these kind of multi-year scales can have really profound impacts on how the growth in soil takes place. Those antecedent conditions can really set up certain fungi to grow well in soil or to grow less well. So one of the things that we've observed with valley fever in California is that this alternation between drought and rainfall conditions can well predict valley fever incidence. So what we're seeing here in this plot is a graph of all reported valley fever cases in California since 2000. And beyond the overall trend of valley fever increasing, you can see that in periods of drought, which is represented by this lighter orange color, we actually see fewer cases. And this is probably because the fungus just doesn't have the moisture it needs to grow. However, in the 2 years following drought, we actually see very large spikes in incidence. And in our ensemble prediction model, we were able to show that these spikes are actually exacerbated by that drought. So to kind of zoom in on a more recent sort of case study, there was a very profound dry wet swing that occurred in recent years. So 2020 to 2022 was an extremely dry period in California, capping off the driest 22-year period since 800 CE. The photo on the right is from our field site in Central California, and you can see just how dry it is. And you can see incidence was relatively low at that time period. However, winter of 2022 was an extremely wet period, characterized by a sequence of atmospheric rivers. Precipitation was 150% of average, and it was one of the top 10 wettest recorded seasons. And you can see just how dramatic California's landscape changed. This is the same field site. Those wet conditions continued a second winter as well. And so this led us to suspect that we would have a major year for valley fever because we had that drought and then we had the extreme rainfall. And so we wanted to get a little bit ahead of the curve. So working with Simon Campanuri, a doctoral student at UC Berkeley, and Dr. Alex Heaney, our moderator here, we adapted our previous ensemble prediction model to enable a forecast for valley fever cases in California. So what you're looking at here in the cyan color is our model fit to the historical data, where the line is our model predictions and the dots are the observed case count. And then in purple was our forecast. And so we were able to forecast 2 years using only meteorological conditions. And then the X's are what was actually observed. So we achieved a pretty good forecast for valley fever in the state of California. And we shared this forecast with California Department of Public Health in advance of the 2024 season, which enabled them to put out press releases to encourage physicians to have increased suspicion for valley fever among people who were showing up with pneumonia-like symptoms. So one kind of outstanding question is, how does prior drought fit into the life cycle? Why is this prior drought important? And one hypothesis that we've been thinking a lot about is how drought interacts with rodents. During drought, there are large die-offs of rodents, and rodent populations dramatically rebound after drought. And if we think of other rodent-borne diseases like hantavirus, we see similar dynamics to valley fever, whereby hantavirus outbreaks are often following droughts when rain returns and rodent populations explode. So rodents are a sort of interesting host for valley fever. We know that rodents harbor coccidioides in their lungs, but we don't really know what else happens. So one compelling hypothesis is that these rodents are harboring these spores inside their lungs without causing disease. And when the rodents die, those spores are able to regerminate and grow mycelia again, this time using that rodent carcass as its primary nutrient source. So one of the things that we've been investigating through some field studies in California is the role of burrows and rodents on the fungal pathogen that causes valley fever, coccidioides. So in 2007, there were these cool experimental plots that were set up in Central California where rodents were excluded from certain areas. So this field site is punctuated by all these rodent precincts that are rodent burrows. And what we have access to are burrows with rodents outside the exclosure zones, and then burrows without rodents inside the exclosure zones. So by sampling a cross-factorial design that crosses burrows and surface soils with rodents and non-rodents, we've been able to try to tease out some of these effects. And what we see, which is shown in the graph on the right, is that about 30% of the burrows that contain rodents have coccidioides in them, but that this percentage drops dramatically to less than 20% once we've excluded those rodents. And the positivity is even lower in surface soils. So this gives us additional evidence that rodents might be playing a key role in mediating these climate processes. I do want to note that these dry-wet swings may be important for other fungal pathogens as well, and is something that we are hoping to study, especially in Michigan, where we don't have coccidioides, but we do have other fungi like blastomyces. So blastomyces also follows perhaps a similar process where the mycelia grow and sporulate, potentially under wet conditions. The act of rainfall may mechanistically jostle these mycelia and liberate those spores, and then dry conditions might contribute to spore dispersal. And blastomyces causes a disease called blastomycosis, which has been increasing in the US in recent years. This is some work done by Julie Bartels, who graduated with her MPH from Berkeley, looking at electronic health record data. And you can see that from 2013 to more recent years, incidence is both increasing in certain states as well as expanding. So I'd like to just acknowledge a whole team of researchers and thank you all for your time here.
Thank you so much, Jennifer. That was a wonderful summary of a lot of, I know, really hard work that you've been doing over the last few years. Okay, let me share this. Okay, so our fifth speaker is Dr. Jane Sykes, who is a professor in the Department of Medicine and Epidemiology at the UC Davis School of Veterinary Medicine. Her research interests are focused on infectious diseases of dogs and cats with public health significance, especially systemic mycoses and leptospirosis. Dr. Sykes, I will hand it over to you.
Thank you very much. Can you hear me and see my slides?
Yes, perfect.
Well, thank you so much to Jennifer Head and Alex Heaney for this opportunity to present on animals as sentinels for studying the impact of environmental factors on fungal diseases. And I see that there's been a question in the chat about this too, so hopefully that goes partway to answering that question. So I want to first start off by defining what a sentinel host species is, other than my dog Bodhi, who's sitting here by Sentinel Point in Bodega Bay. He's very cute. He's also behind me on the bed. So sentinel species are animals that are used to detect risks to humans by providing advance warning of a danger. And some animals can act as sentinels because they may be more susceptible or have a greater exposure to a particular hazard than humans in the same environment. And people have long observed animals for signs of impending hazards or evidence of environmental threats. And so you probably are aware, or some of you are probably aware of the classic sentinel animal in the sort of history of sentinels, and that is the canary in the coal mine. And actually the idea of placing a warm-blooded animal in a mine to detect carbon monoxide was first proposed by John Scott Haldane in 1895, and canaries were used as early as 1896. So I love history of infectious diseases and diseases, so I thought I would just add that one to the conversation. So there's also been some efforts to really define what a good sentinel host species is. And so listed here are some of the characteristics of a good sentinel. So ideally, that animal sentinel should be abundant across time and space where humans are affected. And we've been talking about social determinants of health. So really those animals should be in proximity to all humans in all walks of life, and including people who are in environments where there might be poor resources, for example. They also should be close to all humans, and there should be limited movement of the sentinel host. And certainly companion animals do travel less than people, although there is some travel of companion animals. They also should be very susceptible either to infection with a detectable antibody response or disease. They should interact with both the pathogen and the target population, ideally have a similar route of infection. And then, as I just already mentioned, they should have some way to be detected, either through detection of antibodies or through illness. They should be easy to collect samples from, and I've certainly had some experiences in my career with less easy sentinel host species, and similar discussions surrounding trying to get samples from rodents with Dr. Head. And then it should be possible to capture or diagnose as many cases as possible. And really, ideally, from a human health standpoint, they should develop infection or illness in advance of human disease so that they can predict impending human disease. And it's great to have sentinels, but unless you're using them, they're no value at all. So we need to be really taking more opportunity to use animals as sentinels, because there's a lot of really, I think, lost information just because the potential for this is underutilized. And so why do companion animals specifically make good sentinels for fungal disease? Well, thinking about what I just went through, they certainly are abundant across time and space where humans are affected. And these are recent estimates from the American Veterinary Medical Association. And so you can see that right now in the United States, the number of dogs and cats that are receiving veterinary care, which is, an underestimate of the total companion animal population, is 131.6 million, which is more than the combined human population of Australia and the United Kingdom. So really a tremendous number of companion animals, and these numbers have been rising in recent years. It's now estimated that more than 60% of households have a pet, and actually globally, it's estimated that more than 50% of households have a pet or have contact with a companion animal. And then we also talked about the characteristics of a good sentinel host species as being having limited movement. We talked about travel, but also susceptible and having a similar route of infection. And so my work has been predominantly in the field of valley fever, coccidioidomycosis. We've had some nice introductions to that with beautiful diagrams, as usual, from Mandy Weaver, animated diagrams of how these spores are inhaled. And it's exactly the same in dogs. And dogs are very, very susceptible to coccidioidomycosis. And we've been seeing increasing numbers of cases in recent years, to the point now where we're actually seeing multiple dogs a week with valley fever in our clinic. And then I also just wanted to highlight this interesting historic article that relates back to what Dr. Head was just talking about rodents. And this is an old publication talking about an outbreak of valley fever in a family from Oklahoma that were visiting a construction or an excavation site in southern Texas in Beeville. And so a bunch of family members went to the site and they were mucking around in the dirt, and multiple family members became ill, and they had a family dog, and the dog was actually extracted from a rodent burrow in the excavation area. And the dog developed respiratory distress, anorexia, and then died a week after this all happened. And so they went to the excavation site and they collected soil samples. And of 8 soil samples collected for culture, 7 random and 1 from the rodent burrow, only the rodent burrow sample tested positive. And there are other examples of where dogs have been sentinel for valley fever. When valley fever was first identified in Washington State in 2013, it was reported that the Washington Animal Disease Diagnostic Laboratory had actually detected 2 cases of valley fever in dogs and a case in a horse several years previously. And I actually was contacted by a veterinarian early in the course of my career at UC Davis who had diagnosed a case in a non-traveled dog from Washington State. And then we subsequently did a study looking at risk factors for valley fever in dogs. It was a case control study of dogs coming into the UC Davis Veterinary Medical Teaching Hospital. And we showed that digging really was a strong risk factor for dogs and increased their risk of being diagnosed with valley fever by 11-fold. And the other thing about this disease, as opposed to other fungal diseases, is that in dogs, it really relies on diagnostic testing using agar gel immunodiffusion, shown here, which is only performed in dogs at a very limited number of diagnostic laboratories. And so that means that up until recently, before these point of care tests started to be used more widely, it's been possible to basically capture all cases of dogs diagnosed with a disease. Another important characteristic of a good sentinel host species situation. And so with the concerns about the spread of valley fever with climate change, which I'm not going to go into because they've already been described well, really we have an opportunity to use dogs as sentinel host species to study this spread. And as some of you on the call will be aware, our previous maps of coccidioidomycosis in humans, shown here from the Centers for Disease Control and Prevention, have incredible gaps in them because of a lack of reporting in certain US states. And so we have just published our work done in collaboration with the University of California at Berkeley and the School of Medicine here at UC Davis and other collaborators, showing the use of nearly 900,000 dog test results over the last 10 years to remap valley fever effectively. And this is a aggregated data over that 10-year period, and we've been able to show increasing spread of involved counties. And you can see that where we do have information in humans, we have similar incidences in dogs, but now we have pictures in these states and counties where there's no previous information available from people. And again, working with UC Berkeley, we were able to get these really lovely incidence curves showing how nicely dog maps with human incidence per 100,000 populations. And we're seeing a little bit of a delay in dog incidence compared with human incidence. This is California here and Arizona here. But we're also studying this in a little bit more detail, and there may be an opportunity using other diagnostic test information to get results before human disease appears. So that's probably more than I have time to talk about, but I just want to thank all of my collaborators and co-workers, including all of the diagnostic laboratories that have been participating in our work.
Fantastic. Thank you so much, Dr. Sykes. It's such a great way of describing the power of using, data from animals, and in this particular case, dogs, to better understand the ecology and dynamics of a human disease as well. So thank you. Okay. Our sixth presenter is Dr. Malia Ireland, Deputy State Public Health Veterinarian and Senior Epidemiologist in the Zoonotic Diseases Unit at the Minnesota Department of Health. Dr. Ireland coordinates statewide human and animal surveillance for fungal infections and also works on rabies and enteric zoonoses. Before entering public health, she practiced small animal medicine for a decade, giving her a unique cross-sector view. Dr. Ireland, thank you so much for joining us.
Thanks for having me, Alex and Jennifer. Super great of you to invite me. Okay. Let's see, I'll just go ahead and start talking because it's just the first slide. Thank you so much for the opportunity to join. I'm just going to give some highlights of endemic fungal disease surveillance in Minnesota. My work is in applied infectious disease, which pragmatically speaking is often performed by state or local public health agencies. We collect data on cases of reportable illness, respond to outbreaks, deliver community outreach and education, and do a lot of other things. So we can have a lot of raw data due to reportable disease rules, but I also like to joke that epi data is dirty data, since it's usually not the result of designed studies and so on. So sometimes you just have to deal with what you get. So, okay, next slide. The endemic mycoses, we've talked about a few of them already, but they are systemic infections with fungal organisms that occur in certain geographic areas, and they're considered endemic to those areas. So the three biggies in North America are blastomycosis, histoplasmosis, and coccidioidomycosis, which we've heard a lot about. They can occur in humans and animals, soil reservoir, transmission is inhalation, lung infections, and dissemination. We got all that already. Next slide. So in Minnesota, blastomycosis is all three of those endemic fungal diseases are reportable, which means the healthcare providers and labs are required to let us know if they diagnose a case or have a positive test result. For animals, only blasto is reportable. So we do enhanced surveillance for blasto and histo, and that consists of getting clinical data and completing exposure interviews. When we get a report that meets set criteria, we gather data on that case, such as demographics, symptoms, diagnostic tests and results, treatment and outcome, previous comorbidities, etc. We also focus on dates of illness onset and healthcare dates because diagnostic delay is really common with fungal infections. Dr. Weaver mentioned that already. After medical record abstraction, we attempt exposure interviews. So we ask about their house, yard, local environment, employment, outdoor activities, where those were done, and we attempt to assign the most likely place that they were exposed, which is often not where they live. So in summary, our goals for each case are to understand their exposure location, time to diagnosis, body systems affected, prior comorbidities, and their outcome. Next slide. So we use this data in a few different ways. On an ongoing basis, we'll be watching for clusters of cases who live in or were exposed close to each other. We provide yearly summaries of our case count data, demographics, case fatality rates, and then residence and exposure locations at the county level on our website. And then we publish more detailed info in peer-reviewed journals as we're able to get to that. Next. So this is a little bit of data that shows some of the recent changes. This graph is Minnesota blasto cases per year since 1999. Human cases in navy and the animal cases are in teal. We get a lot more animal cases than people, which, like as Dr. Sykes was just mentioning, animals can be useful sentinels. And here in Minnesota, we have found that they're very useful for establishing kind of the endemic range, the more endemic areas versus less endemic areas in Minnesota, and they are less useful as sentinels for outbreaks. But that's a different topic. Anyway, median case counts for like all of these years for people were 34, animals was 73. But you can see that starting about 2018, those numbers really started increasing. Next slide. Oops, sorry, you can actually go back one. I lied. Yeah, that one. So thank you. Starting about that year, we really see a lot of increases. The median case counts jumped dramatically. It's 292 for animals right now. And 2019, 2020, 2021 were all really wet years in Minnesota, just kind of generally. But 2022 was a really dry year. And so as Dr. Head mentioned, there's a lot of things that potentially we can sort out looking at that climate data with those. Okay, go ahead, next one, Alex. And I think this is the one I'm going to skip for time, so you can go to the next one. So I mentioned that we tried to assign exposure locations. So the map on the left is kind of just the broad ecological regions in Minnesota. And on the right is our case count map for human cases by where they were exposed. So we see that the main areas where blasto occurs, which is the darker green and medium green areas, are really overlap with the zone that is Laurentian mixed forest. So there's lots of conifers, maples, birches in that area, and lots of lakes and rivers. And then we see a lot less blasto in the areas of the state where there's more prairie. And then next slide. That lets us actually compare the other endemic fungal disease that we have in the soil in Minnesota that we do surveillance for, which is histoplasmosis. And Blasto's on the left, it's blue now rather than green. Sorry about that. And then histo's on the right in the green. And they are just kind of nice mirror images of each other along a diagonal there. I think this, we know that they likely have different ecological niches, but I think that Minnesota might give us a little bit of a unique opportunity to explore that even more. So next slide. So there are definitely a lot of gaps in fungal surveillance. As Dr. Sykes mentioned, there's a limited number of states that report fungal diseases. Until last year, there were only 5 states in the US reporting blastomycosis. And a few more states have been added, so it's 8 now, but they're just, it's going to take a while before those states can add anything significant to the data. There are no feasible preventive measures. As I think it was Dr. Reaver mentioned, it's not feasible to wear an N95 outside all the time. People are exposed to blasto in Minnesota doing enjoyable routine outdoor activities. We can't tell people to not go hiking. So that's frustrating. Outbreaks are often detected after the fact because the incubation periods are long. Like, we just don't see that these outbreaks are happening until it is done. Another potential gap is that skills and resources of state and local health departments who may be collecting data on this may not extend to the types of analysis that could be beneficial, especially for these endemic fungal pathogens where the changes, they're not spread person to person. So it's really more about the environment. And that's not something that like a regular epidemiologist, an applied epidemiologist might have the resources to look into. But I think that better knowledge of environmental factors that might be increasing risk would be really helpful. So next slide. So that does bring me kind of to the end, which is spatial temporal analysis and ecological niche modeling may be able to give us more information so we could try to do what has already happened in California for cocci, like good modeling to actually be able to say, this is going to be a bad year, or this particular area is riskier than that area over there. So I have a few different partnerships with scientists, and so we're working on mapping and modeling blasto and hopefully histo, and we're using human and animal case data and exposure location to identify ecological niche, predict weather and soil events that might lead to increased transmission, and then hopefully predict how climate change might impact endemicity and like the changes over time. So that's it. Thank you so much. Oh, and those are my broad-based budding cookies. I just always like to put them at the end of the slides.
Wonderful. Thank you so much. And I think in particular, I'm hoping we will be able to get more into what you've introduced there really well, which is the sort of ideal way for researchers and public health practitioners to collaborate and sort of push forward our abilities to mitigate and prevent future disease burden. So thank you so much. Okay. So our final panelist today is Dr. Mitsuru Toda, who's the Senior Advisor for Readiness and Response and Acting Epidemiology Team Lead in the CDC's Mycotic Diseases Branch. Dr. Toda previously led the Outbreaks and Endemics Unit, providing subject matter expertise on blastomycosis, coccidioidomycosis, and histoplasmosis. She's an alumna of the CDC's Epidemic Intelligence Service, holds a PhD in infectious disease epidemiology from Nagasaki University, and an MS in health policy and management from Harvard School of Public Health. Dr. Toda, I will hand it over to you. And also, would you like to share your screen, or should I share your slides?
It would be great if you could share them.
Perfect.
Great. Well, thank you so much for having me today. Today I'm just going to give a few examples of how we've been observing and seeing the impacts of environmental factors on fungal disease. But we do work on a large portfolio of things at CDC's Mycotic Diseases branch, since we're one of the only public health institutions that focus on fungal epidemiology. Next, please. And next. This has already been discussed by previous panelists, but the frequency and intensity of the extreme weather events are expected to increase as environmental changes happen. And studies like this in California show that coccidioidomycosis outbreaks among firefighters that are exposed to dust and smoke are happening more frequently as they fight the wildfires. Next, please. This was a study that we did in Texas after Hurricane Harvey. But as mentioned, excessive moisture from heavy rain and flooding created conditions that's suitable for indoor mold growth, and associated respiratory tract symptoms and invasive mold infections were observed. So in order to understand whether there is an actual increase in these invasive mold infections, we led a study with multiple institutions to extract medical charts, and we did see that the visits for invasive mold infections increased after this particular hurricane. Next, please. And in Missouri, back in 2011, tornado victims were found to be infected with fungal infections resulting from lacerations that they received when encountering airborne material contaminated with soil or debris. Next, please. And similar traumatic inoculations are observed domestically as well. These are chromoblastomycosis cases that were seen in Texas after a hurricane. These patients lived in rural areas and reported cleaning brush and fallen trees on their properties after they've been disturbed by a hurricane. Please. And also, it's been noted by other panelists, but we are seeing expansion of the habitat of the fungi. Since most fungi are in the environment, greater areas in the US, like what we're seeing with coccidioidomycosis, might become more suitable because of changes in temperature, precipitation, or soil characteristics. Where we've also seen in recent years interesting blastomycosis outbreaks that's happening in areas that that is endemic, but we didn't expect to see in, for example, winter months and things like that. And also the habitats and the range of environments that the host might live might also change. Like, for example, for histoplasmosis, the bat habitat seems to be changing. And so we are seeing more reported cases of histoplasmosis after exposure to bats, like in caves and things like that. Next, please. And this has also been noted, but the environmental changes are expected to lead to more frequent and concentrated applications of agricultural fungicides. And these fungicides can be linked to development of azole-resistant Aspergillus fumigatus. And some of the fungicides that are used share chemically similar modes of action to the antifungal therapies that we use for humans. Patients who have no exposure to, for example, some of these azole antifungals might inhale resistant azole-resistant Aspergillus fumigatus from the environment. So this has become an increasing issue, especially in Europe, but we're also observing it in the United States as well. Next, please. So yeah, that was a quick summary of things that we've been observing here. So hopefully I've been able to summarize quickly how how environmental pressures can affect fungal epidemiology and infections. And also we're increasingly being aware that, fungal pathogens are becoming resistant. And so with only a few classes of antifungal drugs that we have at our disposals, it's really important to really increase our awareness about antifungal stewardship and education about fungal diseases. And so these last slides, I'm just showing some of the resources that we have on our website. And then the next two slides are about Fungal Disease Awareness Week. We do this annually, and this year it's going to be in September. So thank you so much.
Wonderful. Well, thank you to all of our amazing panelists. That was such a great overview of all of the amazing work and action that's being done in this space. So I have seen there's been a lot of great questions in the chat. And so now I'm going to pass it over to Jennifer to facilitate some of these questions and get some answers from our panelists.
Awesome. Thank you so much. Yes, thank you so much for all these questions in the chat. We'll try to get to all of them. If not, we can try to follow up separately for those that we don't have time to. I'm going to start with this question from Leandra, who asked if there were to be an increase in pathogenic fungal species due perhaps to more species tolerating higher temperatures, could it cause a nationwide or perhaps global epidemic as antifungals become increasingly ineffective? So perhaps a question that we've thought of a lot with with the rise of the Last of Us. So I'll start with Dr. Gusa, and then anyone else who wants to add, and please.
Hi, yeah, thank you for your question. This is something I spent a lot of time thinking about. And certainly we don't have any direct evidence yet really tying the emergence of increased disease to temperature, like a thermal adaptation. You're probably aware of, Candida auris is often cited. There's a hypothesis out there, but we actually don't have any sort of direct evidence linking that because it appeared simultaneously around the planet. So there was thought that it was actually sort of a temperature pressure, but there's some debate over whether or not there were already existing Candida auris that were already adapted to higher temperature and able to cause disease. So that's one of the things that we're kind of looking at directly in our laboratory is if we actually adapt less thermal tolerant species to a higher temperature, can you actually see an effect on increase in virulence? So we'd love to be able to test that directly, but that's harder to do. But what I would say is that it's likely that as we are definitely putting selective pressure on fungi to adapt to higher temperatures, and so it does increase the potential. However, any mutation that occurs that allows an organism to grow better at higher temperature could also result in a fitness decrease. And so we really don't know the impact of whatever that particular mutation is that's giving it the thermal adaptation, whether or not that would be something that would also cause it to be less fit to cause infection. And what I'll also say is that because these any particular fungi that adapted a random mutation in the environment, it wouldn't really spread that quickly, because we're not, a lot of these fungal infections are not spread person to person transmission. So it would have to be sort of like a wide ranging event over a lot of different fungi developing the same mutation that also increase virulence. But a lot of this comes back to the host, the actual human host themselves and how susceptible they are. So I think that that threat, while it's there and we need to keep an eye on it, I don't know that it would result in a widespread fungal pandemic. You'd have to have a lot of events happening, but it certainly is concerning that we're putting this selective pressure. And so that's just my initial take, but we'll have to see what evolves.
Awesome. Thank you so much. Here's a question from Deborah that I think is well suited for Dr. Ireland and Toda. What are some ways that we could influence fungal disease surveillance policy at the state and federal levels?
I'll try and jump in. I think kind of approaching that a couple of ways. Just one direct thing you could do is find out what does it take to get a disease added to the reportable disease list in your state. And so that's something where you could reach out to the state health department and find out how are things added to the reportable disease list, like, and how can we lobby for such things. In Minnesota, there's a whole thing. It has to go through statute and get approved by the legislature and so on. So it's not necessarily an easy process, but that would be a direct thing. And then just generally, it's just a challenging time for public health. So I hate to say, call your representatives for funding, but if that's something that you think should have more funding, I think that's something to communicate to people who make those decisions. But I'll let Dr. Toda talk more about maybe the federal level if she wants.
Well, yeah, it's a challenging topic. I know there's so many competing priorities, especially at the state public health level. And so there's nothing that we want to really push from our side, but we have seen champions make this happen in the past few years. For example, Vermont, there was a fellow who worked on an analysis to look at blastomycosis prevalence in the state, and that got people's attention, and I think now it's reportable. So it's great to have those champions. But when I started at CDC, it blew my mind that they're nationally notifiable diseases, but it's not reportable at the state level. So it really comes down to what the state's priorities are. And I think trying to really encourage these champions might be one way to gain success.
Thank you. That's great. Here's a question that I'll punt to Dr. Sykes, and then Dr. Ireland, if you want to comment as well. How might ethics play a role in the use of animals, especially companion animals, as sentinels for fungal diseases?
Yeah, I can maybe make some comments on some of the challenges that we experienced relating to the mapping project, and just being able to get accurate location data. And so obtaining laboratory data, we were very fortunate, and then we had all the laboratories, all 7 laboratories that did the testing, they all contributed data. But for some big laboratories, because of their confidentiality policies, that there was granular information that was hidden. So for many cases, we were only able to get to a veterinary practice level rather than to owner location level. Now, for most companion animals, there was actually one paper that was published that studied how far people live from their veterinary practices in general, and it's actually usually only about 2 to 5 miles away. So much of the data is probably pinpointing to a 2 to 5 mile radius around those veterinary practices. But there was certainly some gaps in our county data that related to confidentiality of information. But you could also argue the opposite, that you're potentially also concealing granular information that could help people who are exposed in their home locations. So that's kind of an interesting conundrum. I'm not sure that we solve it. But from the other aspect, you could be hiding some really important data about human exposure in a very focused area. And we know for valley fever, because of some of the work that the great people have done on this call, that valley fever is very focal, right? You can move from one spot to another, and there can be detectable spores in the soil in one area, and you can just be a few feet away and there's nothing there. So I think maybe those are the comments that I can add surrounding that.
Thank you. Here's a question from Ashlyn about antifungals. Regarding the potential use Regarding the potential for agricultural use of antifungals, are there current effective alternatives to the use of antifungals in agriculture? Yeah, go for it, Doctor.
I can jump in a bit on this. So yeah, I mean, alternatives would be, I mean, you would need basically antifungals in agriculture because, you know, it's really the number one pathogen and the number one threat to food security. But of course, if you think about antifungals, we have, you know, many different classes of antifungals, and many of them, because fungi are so similar to us humans, right? Many of them can't be used in humans, right? Because of adverse events and because they are not specific enough for, you know, for some fungal enzymes versus similar enzymes we humans may have. That's where, you know, obviously I think there are definitely options in looking into antifungal classes that can't be used for treating human disease, but could still be feasible for treating fungal disease in plants, basically. So that's definitely one way to look into that. And yeah, we need, I think, effective antifungals. And if you talk to farmers, so I mean, it's such a essential part. I don't think there's an alternative that will kind of like, you know, but maybe others jump in. I see Jane Sykes has her hand up.
Yeah, thank you. I can probably make some comments on this because I just gave a presentation on it. And do you mind if I just share a couple of slides on this?
Go for it.
Because I think that these kind of illustrate the problem. And the main azoles that have been used in agriculture are tebuconazole and propiconazole. And so, and you can see just the rise in azole use, and you can see that it's a multi-billion-dollar issue here in terms of the protection that results from agricultural application of fungicides. And it's estimated that 1/5 of global food crops actually die in the field annually due to fungal disease, and another 1/10 rot in storage. So it's not really a very easy thing to tell people to stop using agricultural fungicides, but there are different options. You can see the triazole use on crops has actually increased 450% between 2006 and 2018. And this is because that these azoles are inexpensive. They're very broad spectrum. And the thing that we don't, as medical healthcare providers, like about this is that they have very high environmental persistence. So that's good for keeping fungi off crops, but it also selects for resistance in important human pathogens like Aspergillus. But there are other alternatives. It's just that triazoles are really appealing because of their properties that are mentioned here.
If I could just jump in real quick to answer that as well. There's some really great non-drug alternatives that might be forging away in the future. If you've heard of RNAi silencing, so spray-induced silencing, Hönigl Jin from University of California at Riverside, she's had success in, essentially what this does is there's certain fungal pathogens that grow on crops that if you introduce RNAs that target their virulence genes or effectors, you can actually shut down their ability to cause disease on plants. And so she's rolled this out on a couple of different crops and had really good success as a sort of a non-drug alternative. So the question then becomes, do people feel this is safe for the crops and in our ingestion? But I think it's a really great alternative.
Well, amazing. Dr. Toder, would you like to add anything?
Oh, yeah, very quickly. CDC has been working with EPA and USDA on this issue to try to preserve medically important antifungals. And there is a framework that I can share in the chat as resource, but for antibiotics, for example, there's an approval process that's in place where if any antibiotics that are going to be approved for animal or plant use that has significance for human medicine, that there's a whole risk and approval process. And we're really trying to work collaboratively to try to push that on the fungal side to see if it could be something that's debated and the risks are clearly understood so that we can preserve those for future use.
I just wanted to bring one more thing in because we are always discussing, we're living in a world where basically all our food or lots of our crops at least have been treated with, fungicides. But this is also a good side, right? Because if you look into other countries, third world, low middle income countries, there's much more problems with mycotoxins, right? So on the other hand, if our food intake contains lots of fungi, fusarium, etc., because we don't use fungicides, right? It's not only causing, the plants to suffer and, the production to suffer, but also food intake is something that's then associated with certain cancers and is unhealthy, right? So I think that's where everything becomes very complicated, and it's just different people coming together, basically, and have to talk about this from different angles. But I agree, alternatives that help us reduce the fungi on crops, help us reduce the mycotoxin intake if we eat them, right? And at the same time, don't cause cross resistance or increasing triazole resistance rates in aspergillus, for example, would be a great solution.
Awesome. Well, that was a very fascinating discussion. Here's a question asking about citizen science efforts and whether they might help us improve fungal disease surveillance efforts. So I'll throw that out for any panelists to jump in. And I suppose while people think, I can come up with, there's a few examples that I can think of that are happening, being led out of CSU, California State University, Bakersfield, where folks specifically through the schools are asked to kind of collect soil samples around their homes and parks and just places they visit, and then it's brought back to CSU Bakersfield and tested for coccidioides. And coccidioides has actually been identified through some of those efforts. I've heard of similar efforts. There was a great study published in PNAS a while back that was called The Fungi of Our Homes, where folks across the US were mailed swabs and asked to swab their door frame. And then they sent those swabs back, and the swabs were analyzed generally by, I think, ITS2 sequencing. So it was looking at a broad array of fungi, not just pathogenic fungi. And that was quite interesting as well. And then there have been some citizen science efforts, you could say, where, at least that I know of in California, where folks who've used filters either for policy purposes of tracking particulate matter or for to filter like smoke that might be coming into their homes on swamp coolers, have donated those filters to laboratories to be tested for specific fungal pathogens. So those have been really helpful in kind of helping folks draw the endemic risk maps for different fungi. Dr. Toda, do you have other examples?
Yeah, I know of examples in Europe with Dr. Matt Fisher, where they did citizen science in the UK, where they placed think sticky pads along the windowsills to look at prevalence of azole-resistant Aspergillus fumigatus there. We are funding partners in Latin America to do something similar. And actually, within our lab, we did something at our homes to try to see if we could observe that. So there's some efforts, but I'd love to see more happening here domestically.
Yeah, I have basically one more, also from Europe. Originating from the UK, not necessarily Matthew Fisher primarily involved because it's a different topic. Moldy houses, basically, and that's something where we see big differences in Europe, interestingly, between different countries. And it's not just income related, it's just certain countries, climate, of course, comes in, but also the general housing, what's the environment of housing. So that's basically, we see strong differences, and that's something where we currently start or at least plan out basically a survey across multiple European countries, citizen science, where we ask, about their homes and about some symptoms. And so that's also, I think it's interesting in Europe because there's so much diversity, right, in a relatively small area with all these different policies in place. We'll see what comes out of that.
I think, Martin, what you might be talking about is the Well Home study. Is that the one you're talking about with Imperial College of London? It's really great. I just came across it, and it's something I'm interested in potentially in looking at these homes that are affected by mold. But they essentially had like 100 different homes in the near Imperial College of London. They enlisted people. They got information on their health backgrounds and like whether or not people had asthma, and they had both passive collections, like just open petri dishes in their homes, and collected them over the course of the year, in addition to like just looking at air pollution. They had detectors in every home. And it's amazing the kind of information we're getting, and it's amazing how limited our knowledge is of the indoor environment and what we're exposed to. But there should be a paper coming out soon on that.
And that's where it builds on. It's not it's a little bit a broader study and a little bit more superficial because it goes over multiple European countries. But that kind of like, I think, the UK and also the Imperial College study is basically, that's the basis of this basically, and why we now try to expand that and look kind of like on a more superficial way because it's much larger, obviously, but expand that beyond the UK borders. But obviously UK is the center of this research, and also, for good reason, because they do have more problems with moldy housing compared to other places. So.
Awesome. Thank you so much. Well, there's a few other really great questions in the chat, but I know we are at time. So I might pass it back to Dr. Heaney to conclude, and we would be happy to follow up with those of you who did not get your questions answered in the chat.
Thank you. Yeah, what a fantastic discussion, and I feel like we could go on for a long time, but we both, Jennifer, on behalf of Jennifer and I, we want to thank you so much to our panelists and to all who joined. We wanted to just leave everyone with sort of a couple key takeaways. The first is that environmental change is already reshaping fungal risk, and we heard a lot about that today with all of the great talks. The second is that interdisciplinary data, so clinical, environmental, veterinary, is essential in us tackling these challenges. And the last is that translating science into diagnostics, surveillance, and preparedness really needs to keep pace with all of these changes that we're observing and studying. So we really appreciate you all taking the time out of your busy day to be here. Please do not hesitate to reach out to me or Jennifer if you have any further questions. And yeah, we hope to hear from you. Thank you so much.